about this sort of thing. On lab, you will have to tell me how a few muscles are named, a couple maybe, okay? And just sort of using these terms that are named for origins and insertions. Parallel muscles, I think that's pretty self-explanatory. Circular muscles, convergent muscles we went over. The pinnate muscles with the oblique fibers, like that. This is how they work. So it's like a transmission. You get a lot of power, but you're in low gear when you have a pinnate muscle kind of configuration like that. I wanted you to remember the aponeurosis, the term, and the fact that the glia, aponeurosis, and the central tendon of the diaphragm are the two that you're responsible for being able to name. Okay. The retinacula of the wrist, there's one just like it around the ankle. We went over that in lab. Remember the histological names. If it's cardiac muscle that you're trying to look at, it's involuntary striated cardiac muscle is the full name. Some people were a little confused on this in lab today, so let's talk about this again. The fascia that surrounds a whole muscle, okay, is called the epimysium. Okay? When you get the different fascicles in a bundle like this, that is the perimysium. Okay? And then when you pull out a fascicle and look at a single muscle cell, which is also called a muscle fiber, it's endomysium that surrounds that. Okay? So there's endomysium around that one, around that one, around that one, but it's perimysium around the whole thing. Okay? Remember those contracting arms, those structures that I drew up on the board there? That's what gives it this banded appearance. Okay? This is where the thin fibers are. The actin, this is the Z line. Okay? And you will be responsible for knowing the names of the bands and the lines. Okay? The actin comes off the Z line and sticks out about that far. Okay? The myosin is a thicker banded. You're wrapping all those walking proteins around each other, and it gets to be a fairly thick band. Okay? So that's from here to here. Okay? So when it overlaps the actin, you get a darker band. Okay? That's why this is a little bit lighter here, because the actin doesn't go that far. Okay? But that's what causes the bands. A band is the dark one, the light band is the actin. You can guess which one of these bands disappears when the muscle contracts.
The I band.
The I band.
Great, the I band, yeah. The myosin is not going to go anywhere. It's just walking arms. It's not going to go anywhere. But it's pulling in the actin, so the Z lines are moving in and the I band is disappearing. Okay? And each one of these things, from that Z line to that Z line, that's called a sarcomere. Okay? The cell membrane, okay, you get a bunch of these are called myofibrils when you put a bunch of sarcomeres together. Okay? Myofibril as opposed to muscle fiber. Okay? That's the cell itself. Okay? That's not a muscle fiber, that's a myofibril. Okay? When you put a bunch of those together inside of a cell, then you've got a muscle fiber, a cell. And the lining of that cell is called the sarcolemma. Sarcolemma, L-E-M-M-A. Okay? And here's your fascicle again. Here's where the perimysium would be wrapped around. Okay? And when you get a bunch of fascicles together, then you have epimysium around the whole muscle. Okay? And it's pretty under the microscope when you look at it. These are your bands. You can see this is one cell right here. Okay? And there's the other border here. So that's from here to here is one cell. From here to here is one cell. So this little thin connective tissue in here, that's the endomysium. There's not much to it. It's just very mild collagen type structure. And then when you put a bunch of cells together, you get the fascicle. Okay? And the fascicle is surrounded by perimysium. Okay? So and when a bunch of fascicles together, then it's epimysium. Okay? Notice these striations, though, how pretty they are, how long these cells are, and the fact that they're usually multinucleated. Okay? Several nucleus along here. It's hard to tell which one it belongs to, but that cell has multiple nuclei because striated skeletal muscle, one cell goes from tendon to tendon. Okay, it's a long cell. So you have to have multiple nuclei along the cell, because that's where you're generating the RNA for making proteins, okay? You can't have a nucleus way up here and get enough protein down here to keep that muscle going. Okay? So you have multiple nuclei that are all in there turning out the proteins that are necessary to make the cell. Cardiac muscle is striated like this, but the cells are only about that big. They're not much bigger than the nucleus, okay? And you'll see a wall where they connect each other. So that's the picture that's going to be coming up here in a second. So you can really tell them apart just under the microscope pretty easily. Let's go to that. All right, right there. Since we're talking about it, let's show you the picture. Okay? It's a few slides ahead. Here's the disc, okay? And it has a name, but this is what divides the cells. And you'll notice that it's not just one cell hooking to another cell. Sometimes they fork and hook to two cells. Okay? There's branches here. That might be a branch there, a branch over here. Okay? So they're not quite regular. It's not as pretty as skeletal muscle, but you can still, especially down here, you can still see the striations. Okay? And that's because those sarcomeres are lined up in banded structures like that. Okay. Smooth muscle looks entirely different. It has the walkers and everything, but they're not lined up to where they band. They're sort of at angles, so you don't get a banded look to them. Okay, I'll show you that in a little bit too. Let's go back here. Okay, things that should be on your word list again: sarcolemma. Don't worry about the triad, but the triad is just where the sarcoplasmic reticulum joins that T-tubule, where the charge jumps down into the cell, with the positive sodium charges. Remember that action potential. The sodium is coming in, and it's going to go down the T-tubule, and as soon as it hits one of those calcium pores that opens up, then the calcium will dump all over the sarcomere, okay, and you'll get a contraction, okay. But this bluish area right here, this is also where the calcium pumps are that suck it back up, okay. So this is where all the calcium is stored, in this blue area. The yellow is the T-tubule, and it's a direct extension of the membrane out here. It's a direct extension of the sarcolemma, okay. So I'll say it again. It's a direct extension of the sarcolemma, okay. Go ahead.
That's not ours.
Oh, it does.
Oh, good. I was looking for something. I must have missed something.
Your book has some good pictures of this too. And is this one in your slide?
Yes.
It's the same thing. It's the same thing. I just thought that this was a little better picture. I put this in later, I think. But it's the same thing. So sarcoplasmic reticulum is where the calcium is stored. Okay. And then the T-tubules, just have that in your vocabulary. Okay.
What did you say was connected directly?
The T-tubule. You can see it has to be, because that sodium charge is marching along this membrane, and then it dives down into the tubule, and this is where it's going to trigger those calcium channels. Okay. And this is the same thing, just another artist rendition. Okay. Here's all now it's green. The sarcoplasmic reticulum is green. The path of the action potential is showing you coming along the membrane, and it's diving down the T-tubules, not just one, but all of them. Okay. That releases the calcium. Okay. That's going to cause all the walkers to start walking. Okay. So again, you have the same terms here: sarcoplasmic reticulum and sarcolemma, sarcomere, from Z line to Z line. Okay. So we're just sort of building a little vocabulary right now. The few questions to see if anything sunk in last time. What generates the sodium-potassium concentration differences?
One more time.
The sodium-potassium pump. Sodium-potassium pump. Okay. That is all that's going to do, though, is generate a concentration difference. It has nothing to do with membrane charges or anything, because they're all balanced. Every sodium out here has a chloride or a phosphate or something that balances the charges. So we haven't got the potential difference yet. What causes that?
The potassium channel leaks out.
The potassium leak.
Yeah.
Right. As soon as a little bit of potassium leaks out, you've got extra positive charge on the outside, and you've left negative charge behind okay, that's unbalanced. So now you've got a potential difference.
And is that what goes down to the minus 70?
Exactly right.
Okay, and that's when the sodium kicks in and brings it back up to the 50?
Well, that's down the line.
That's down the line.
But why does it stop at minus 70? Why doesn't the potassium just keep leaking out?
Because that's when it...
Sorry.
Let's say it's equilibrium.
Well, it reaches an equilibrium, but why? As far as potassium is concerned, it's going down a concentration gradient. It could just leak forever.
Doesn't it get
Because
stopped?
because
Doesn't it
the
get stopped?
The sodium, the little gates block.
The voltage.
The voltage.
Once you get enough negative charge down below, the very last potassium's going, I want to get out, but man, they're holding me back.
That's right.
Because that negative charge is attracting that potassium. And it takes a minus 70 millivolt to do that. And minus 50 isn't enough to do that. Minus 90 is too much, and that's just the way nature does it, okay? But at about minus 70 millivolts, there's enough negative charge right underneath the membrane to hold that last potassium from getting out, okay? And then we call that equilibrium. And now we've got a potential, okay? That's how it's formed. Okay. And then, now we talk about the sodium channels. Now we want to play around with that membrane. We want to do things with it. This is what's different about muscle and nerve cells. Other cells can't do this. They don't have those sodium channels that are chemically gated. You know?
Okay.
They can't fire a potential. Okay. But so the nerve, if you remember, the axon, because it's holding on to the nerve, the action potential is going to be coming down the nerve, and it's going to be sodium positive charge, just like in the muscle, okay, until it hits a calcium channel, a voltage-gated calcium channel. It lets calcium in. That triggers the vesicles to release the acetylcholine, okay? The acetylcholine is now in the synapse, okay? That's going to go on the acetylcholine receptors, which is a chemically gated sodium channel, okay? They're going to open up and dump sodium in. It's called depolarization, okay? Because the membrane is resting as polarized, okay? Minus negative, it's polarized. So when you change that, you depolarize it, okay? So that's what we say, when the sodium rushes in, it depolarizes the membrane, makes everything more positive underneath. If enough of that happens, and it gets out around the neuromuscular junction, okay, because these chemically gated things are only right there where the nerve is. They're not out here along the nerve, okay? They're right there, are along the muscle. They're right here where the nerve is. And if you get enough positive charge flowing out from that pool, it's going to hit the voltage-gated sodium channels. Then they're going to start dumping stuff in, and they're going to excite all their brothers and sisters down the road, okay? So that's how that positive charge spreads out like a wave, okay? And then we talked about how behind the wave, everything goes back to normal because the channels are closing, okay? So what happens when you see this voltage change, this is called the action potential. Right about here at threshold, if you get enough acetylcholine, if you don't get enough, you won't get a fire, okay? But if you reach threshold and get enough positive charge in there, they will fire, the voltage ones will open, and it'll depolarize until you get up here, get this positive 40. This is the voltage that turns off the sodium channel. And it's right about in here, similar voltages that turn on the potassium channels, so that now potassium can flow out, not leak out, but flow out, and you get that big jump back to negative in behind the membrane. Okay? And it even goes too far. This is called the refractory period. This is where the nerve or muscle can't fire at all, because the membrane is too far. It's too negative, and nothing can get it back up to this threshold at that point. Okay? So that's, and then the sodium-potassium pump and the leak eventually say everything's back to normal, and they work out the differences again and come back to that minus 70. Okay?
So you say when it goes up to 40, it turns off the sodium?
Let's do this next one here.
Oh,
I've got
okay.
one for you. I think you've got this slide.
Okay.
Yeah. So, and I might show you an action potential, and I'll put an X right here and say, what's happening at X? And then I'll give you choices, okay? Potassium's turning off, sodium's turning on, blah, 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 something like that. I want you to understand what's going on, okay? So this is where the sodium channels open. You get enough voltage, the potassium channels begin to open and the sodium channels close, and everything starts to go back negative. And then the potassium channel closes. It's now it's no longer flowing. It's now only the leaking channels are working. Okay, so it goes back. Okay, sodium potassium pump is active here, and all the excess stuff either diffuses away or it gets rebalanced by the pump. Okay, is that
Thank you.
fairly clear this time?
Can, and the
I know this
sodium,
is tough to get.
I'm sorry.
Go ahead.
The sodium creates a positive charge, and then when the potassium,
Underneath the membrane.
yeah, underneath the
Yeah,
membrane,
yeah.
it's positive, and then when the potassium
Leaves.
leaves, it's a negative.
Yeah, because
Okay.
it's leaving negative behind.
Yes.
Okay?
Okay.
You're actually losing positive charge to the outside, leaving negative behind, and that's why the voltage goes down.
Okay.
But that's only if you're reading the voltage on the underneath the membrane. Okay? If you're reading the voltage on the other side, it's going to be different. Okay.
Can you refresh us as to at what point that turns that calcium on?
This has nothing to do with calcium here.
No, but when it goes, isn't this the process of it getting to the
Yes,
T-tubules?
yes, this is what's going to happen. This wave, this voltage wave is going to go all the way down the membrane, and it's going to go down the T-tubules
Okay.
until that sodium that's rushing in, causing positive charge, turns on the calcium channel.
So this just keeps going as long as it needs to until it hits that,
Yeah,
and
yeah.
that's when it does its magic.
Right,
Okay.
right. Yeah, you'll get one wave that goes across the cell
Okay.
because it's going back to normal behind it.
Okay.
Okay, and if there weren't T-tubules, it would just stop.
Okay.
Okay, but this is actually how they're sending the message down into the cell,
Okay.
going down the T-tubules. So,
And that just keeps kind of happening over and over again until it hits that T-tubule.
you'll get as many action potentials as you get acetylcholine dumps.
Okay.
Okay, so every time the nerve vesicle dumps acetylcholine into the synapse, you're going to get an action potential.
Okay.
So if you want to keep that nerve, that muscle firing and contracting, you keep stimulating it.
Okay.
Okay.
The acetylcholine is what stimulates this.
In the neuromuscular junction, yeah, where the nerve is dumping the acetylcholine on those
And that causes...
chemically gated channels. Okay, and they may or may not. If we go back to this one, if they don't dump enough acetylcholine, you may get some of those chemically gated sodium channels to open, but they may not open enough of them to go positive enough to fire the voltage gated ones.
Okay.
Okay,
So if you don't, what happens? Nothing happens?
nothing happens. It just, the acetylcholine is eaten up by the acetylcholinesterase, and all the sodium potassium pump just takes everything back to normal. Okay.
So what did you say about the refractory period? What happens there?
It's just sort of an overshoot. You've let out too much potassium, so you get too negative. Okay. And then it takes a little while for the cell, and I mean a very little while. Notice these are milliseconds. These are milliseconds, okay? So it's a very little while before those pumps get everything back to normal. Because they're the potassium pump. The potassium pump opened up here, so it's letting out a lot of potassium, and that's leaving negative charge behind. That's why we start reading a negative voltage, okay? But it overshoots. It lets out a little too much potassium, and it takes a little while for it to come back to normal. And this is the wave, though, that has to get here. It has to get to the T-tubule, the action potential. It drives down in here. That sodium is still rushing in. And as soon as it hits one of those calcium-sensitive channels, or the voltage-sensitive calcium channels, then all the calcium in here is going to be dumped onto the myofibril. Okay? So calcium is the last thing that has to happen, but it has nothing to do with the action potential. Okay?
And then it just gets sucked back up into
Yeah.
where it was, and then it just does its job again.
Right, by the ATPase pumps on here that pump the calcium back into this sacs.
Okay.
Okay, and that's what stops the contraction.
Okay.
So if you want to keep that muscle contracting, you have to keep dumping acetylcholine on it to keep that action potential keep diving down into the T-tubule.
Okay.
And that just keeps giving more and more calcium to the muscle, and it gets stronger and stronger and stronger, and it contracts more and more and more.
Okay.
And it's amazing how finely we can control that. I mean, considering how fast it happens and things like that. And the amount of nerves that are needed to control these things. Your facial muscles of facial expression, as well as your tongue and your hand, take up as much of your motor space in your brain as the whole rest of your body. Okay, which means that there are a lot of nerves controlling the muscles in your hand. Okay. Which means that one nerve controls maybe 4 or 5 muscle fibers, okay, makes them fire. So it's very finely tuned, and in your eyes, it's the finest tune there is. You have very few muscle fibers that are controlled by one nerve, so that you can move your eyes with very good accuracy and carefulness, I guess, is what you can do. But there might be hundreds of muscle fibers controlled by one nerve in my thigh, because I don't need that kind of control in my vastus lateralis. Okay, but in your eyes, you need a lot of control, so there's a lot of nerves controlling that. And just over time, we've developed the same kind of control in our hands and fingers. Okay, a lot more control than you have in your foot, say. Okay. Okay. And I just threw this up there so you can have the words. Okay. I'll probably give you a picture just like this and ask you to name some of the bands or the lines or whatever. Okay. So I don't think we need to spend too much time. I'd like you to have passing familiarity with two proteins because I think they're really interesting. Dystrophin is the protein that holds the Z-line to the membrane, okay, to the sarcolemma. Okay. And they just found that out recently. It was back in '86 after they discovered the gene, okay. And then the guy, it's now still the longest gene known. It's one of the bigger proteins, but it has the longest gene on the DNA known. Almost 2.5 million bases. 2.5 million A's and T's and C's and G's. Okay? It's a huge gene. But look what happens. It takes 16 hours to transcribe it, to make the RNA. Okay? And then the 79 exons need to be cut out, you know? Or should I say 79 introns need to be cut out before they are all hooked back together. Remember, the bad part of the gene is looped up and the good part is hooked and the bad part leaves? 79 times, and it all has to work perfectly to get the right protein to hook that to Z-line to the membrane. So that's kind of weird, you know? That's fantastic. But when it goes bad, it's why we have so many different types of muscular dystrophy, okay? Duchenne's muscular dystrophy is a problem with dystrophin, and it's an inherited form of the gene. Okay? So this is the Jerry Lewis Peloton thing, muscular dystrophy. Okay? But you can see there's so many proteins that make up this whole sarcomere type structure that lots of things can go bad. There are hundreds of muscular diseases out there. Okay? Look, this one here, the other one I want to show you is titin. This is actually the largest known protein. Okay? 34,350 amino acids in the protein. Okay? Incredible. So that's got to be a pretty big gene too, but it's certainly not 2.5 million. Okay? And over 400 mutations already known and characterized by genetic studies on these problems, on these proteins. But what titin is a spring. Okay? When these walkers walk, they pull the Z-line in. Okay? When everything's done, the feet let go, and then you got this big spring that pushes the Z-lines back out. Okay? That's what titin does. It's a spring. It's a protein that's coiled up and compressed when you're contracting, and then there's mechanical energy there now. And so when you're resting, it springs back and opens up the Z-lines. So, two real interesting proteins, I think. Don't worry about these numbers, though. Okay? But just kind of have a basic idea of what dystrophin and titin are. This is the video I think I showed you. This is what it actually looks like. This is the neuromuscular junction on an electron microscope. You can see the little flip-floppy areas here. This is the motor implate on the muscle. Okay. This is the synapse, the space in here. You can see the vesicles of acetylcholine here, the little round vesicles. Okay. That's what's going to be dumping acetylcholine into the cleft here. The dark areas, this is the acetylcholine receptors. Okay. This is where all the sodium is going to be rushing in. So this whole area down in here is going to build up that positive charge along the membranes. And if it gets out here far enough to hit the voltage-gated sodium channels, then you're going to get an action potential that fires along the muscle membrane. Okay? But, pretty fascinating little thing. But so this is, like I say, the muscle part is called the motor end plate. The space is called the synapse. When you put the axon with it, the whole thing is called the neuromuscular junction. Okay? Clear enough? And I think this is where I was going to compare the striations again. Don't worry about identifying this kind of stuff. But you can see this is a muscle cell, so this would be a muscle fiber. This would be endomysium around it here, perimysium around the fascicles. And you put a bunch of these fascicles together, and around the whole thing would be the epimysium. Okay? We'll talk about spindle fibers when we get to the neuro section. Don't worry about that. Those discs, I showed you the lines here. That's where the cells meet. Okay? What they actually are is basically walls and walls with gap junctions. Okay? So you've got calcium that can move back and forth through these cells and get the next cell to fire in unison or in rhythm with it. This one is going to fire a little faster than that one does, and that one's going to fire a little faster than that one does. So you're going to get rhythmic contractions, and that's how you get a heartbeat. Okay. This actually might be. I'm not going to. Don't worry about being tested on it. But why do you think the heart can beat on its own? What triggers it? It doesn't need nerve. It doesn't need nerves.
Well, is it because of the calcium going through those gap junctions that?
It's the blood pushing.
Well, it would just never stop.
It's the electrical.
Oh, okay.
It's the electrical. The electrical.
Is it not the way the blood flows through the heart?
I'm sorry.
The way, like, the blood flows through it, it keeps it.
No, that's just a function of what the heart does. It's a pump. You know. It's interesting. It's a pump that keeps itself alive by pumping blood through the cardiac arteries. It's interesting. Okay? But you can look at the heart like any kind of a pump. It has a mechanical valve, a mechanical force. It has valves, and it has an electrical system. Okay? So it's like a motor. Okay? But the electrical system is interesting. You can cut off the vagus nerve, the cranial nerve that controls the heart rate. When you get frightened, the vagus nerve, fires these things. That can actually slow down the heart rate. But the adrenal system, the adrenaline that's in your system, fires up the heart rate. Okay. But if you want to slow down the heart rate, you can massage the carotid. If you get palpitations, just massage the right carotid, and that massages the carotid body there, and the vagus nerve will slow the heart rate down. So sometimes you can do that in the ER. Somebody comes in, you just rub their neck, and it's gone. But you can get away without any of that, adrenaline or vagus nerve or whatever, and that heart is still going to pump on its own. Okay. The reason is I didn't expect you to know. The reason is kind of tricky. We've got a calcium leak, not just a potassium leak. Okay. Potassium leak gives us that negative charge underneath, but a calcium leak lets calcium come in, which is positive, so you get to threshold because of that calcium leak, and then you get the action potential. So it just does it over and over again. It fires itself by leaking calcium into the cell. Okay. And there's special cells in what's called the sinoatrial node and the ventricular node, special cells that do that. And that's sort of the electrical system of the heart. Okay? That's the part of the heart you're trying to, when you give it a shock, you're trying to get it going. What's that?
The ionized calcium, does that kind of?
Iodine, well, yeah, it's an ion.
Yeah.
Calcium is 2+. Yeah. So those heart muscles have a special protein membrane pore that leaks calcium in, and over a certain amount of time, within a second, the calcium leak will cause that voltage to go positive and reach threshold, so it fires the heart. Okay? So that's why your heart will beat by itself. And all we do with drugs is try to speed up these processes or slow them down. The vagus nerve tries to slow it down with acetylcholine. Okay, it's
So the amount that your heart beats is how fast the calcium is dripping?
yeah. And you can actually have people that have problems with that whole firing issue, and they'll have rapid palpitations. They can be born with heart rates that are pretty high or pretty low. I mean, some people are born with heart rates of 35, 40. Okay, and that's because they're born with that kind of problem. But a normal heart beats 60 to 80 beats a minute. Okay, and that's only because you're getting a little bit of tonic stimulation. If you turn it all off, the heart will beat by itself about 40 beats a minute. Okay, So then just the fact that we're beating at 60 to 80 normally means that we're stimulating the heart all the time a little bit, okay? But you cut that off and the heart will start beating about 40. That's when people need pacemakers, okay? That's usually why you put pacemakers in, because their heart rate is too slow, okay? They cut off that system. So you just turn it up a little bit, get them beating around 60, and that keeps them better.
Dr. Hurts, I have a question.
Yeah.
So what about the professional athletes that have like Lance Armstrong, his heart beats are like 35 beats a minute?
That's a matter of extreme conditioning. Yeah. You got a heart muscle that's so big, and his heart is huge. Okay, it takes up a lot of his chest. And you just don't need a fast heart rate to have the same cardiac output as another heart. Okay, so when you get a big, strong, muscular heart, it can beat, just once, and it's pumping out 3 times as much blood as the next heart. So it can beat 3 times slower. Okay? That's the only thing. It's the cardiac output that's important. That's what's keeping all the other organs alive. Okay? But where that extreme conditioning comes in is when you need a lot of extra oxygen and nutrients in your system to run the muscles in extreme activity, and then the big heart helps.
But that's different than an enlarged heart.
That's different than a heart that's being exercised by high blood pressure. Yeah, you have high blood pressure, and the heart, a normal heart, has to push against that pressure, and it gets bigger just like any other muscle. But it's not really conditioning right. It's like, oh please, I'm not ready for this, I can't do this. But it keeps growing and keeps growing and keeps growing until the point where it just says, I can't do it anymore. And that's when you get congestive heart failure.
So what about a person that has a hole in their heart? Does that affect the pumping in it?
Yeah, it affects the efficiency of the pump. If you've got one chamber that's leaking to the next chamber, you're not getting the blood out that you should, because it's going back. And you're sort of having to pump a certain percentage twice, okay? So it's not efficient. Atrial septal defects between the heart, and valves that aren't working right, or valves that are too narrow, that don't let enough blood get out, That's why you hear heart murmurs when you hear that and the blood is squirting through those valves. Yeah, there's, like I say, the things can go wrong with the pump, with the valves, and the electrical system. It's just like a motor.
So just to clarify, so the heart does not rely on outside nerve signals, right?
Right.
It has its own nervous system.
To
pretty
be
much.
like skeletal muscles, they would never fire on their own. But the heart has that strange
its own
calcium
thing.
leak, and that's what causes a regular contraction. Okay?
Yeah.
Here's the difference when you look at it under the microscope. Skeletal muscle, nice long cells, multinucleated, okay? Cardiac muscle is mononucleated, and you see the intercalated disc separating the cells, okay? So it's not quite as linear and pretty as skeletal muscles. So I think you won't have too much trouble differentiating these two. And we have that poster in the lab that shows you the pictures, and we'll probably put that out and say, what is B? Is it cardiac or skeletal, smooth muscle? Okay.
And it's both skeletal and cardiac that are striated, but smooth
Striated.
muscle is not.
Right. And here's smooth muscle.
Okay.
You don't see any striations. Okay.
Looks like the skin
That's-
slide you're showing us.
This looks a lot like connective tissue, doesn't it? It looks a
Yeah.
lot like tendon.
Yeah.
Okay. And these are the nuclei, the dark ones there. So the cell is only about that big.
Okay.
Okay. They're not huge. You line up lots of them, and this one is going up and down. This is a longitudinal muscle. This is a circular muscle. This is smooth muscle here that's coming out at you, because this is the lining of the intestine, and the esophagus would look very similar. The stomach would look similar. You could look at the layers. What you've got in the intestine is smooth muscles that line it horizontally, let's say, all along the edges, okay? And then you have a donut kind of wrap of smooth muscle all around it as well. So you have two separate types of fibers. You've got fibers that are just going to constrict, and then you've got fibers that are going to shorten the intestine.
Okay.
And that's how you get things to move along.
Okay.
Okay? So a peristaltic wave that goes down involves a squeezing and then a shortening, and it leaves the new bolus further down. Then you get a squeeze and a shortening, and the bolus keeps moving.
Okay.
That's a peristaltic wave for the GI tract. And it has its own brain. It has its own waves too. It has little neural ganglia that stimulate it. I forget the exact timing of the waves, but it's like there's one that occurs every 5 minutes, and one every 8 minutes, and one every 15 minutes, and they're different strengths of contractions. And so the intestinal system has its own brain. We don't have to control it. We don't have to tell it to move things on. It senses the pressure, it senses the pH and the sensitivity and kind of what's in there, and it just moves things on. Okay, it's at its own little enteric nervous system is what it's called. Okay, we can speed it up with our vagus nerve, and when we're hungry or when we see food, our brain stimulates the vagus nerve. That stimulates the stomach and the intestinal tract to start contracting a little bit. It stimulates the acid production in the stomach. And so we're getting ready. It stimulates the salivary glands, so we start salivating. That's all the parasympathetic nervous system, the rest and digest system. It's the opposite system to the fight and flight. Okay, so, but it will contract on its own even if you cut the vagus.
Hopefully this doesn't bring us too off track, but what is it that causes, like, you have something to eat and then like an hour later, people say, didn't agree with me. What is that,
Who knows?
Okay, but it is something just...
gallbladder problems or ulcer or just, yeah.
And then,
or maybe they just, spicy food is too hot for them or whatever. People, it's all kinds of human variations.
I just didn't know if it was
Food
just your
tolerance.
muscles,
Yeah, no, it really has not too much to do with
not
that.
to do that.
The common thing you'll see with the GI system as far as pathology is in diabetics, you get something called gastroparesis. The nervous stimulation to the stomach starts to go, just like the nervous feeling in their feet starts to go. When the diabetes starts to kill nerves, the GI system slows down. Okay, so you get gastroparesis, and the stomach might stay full for 10 hours instead of 3. Okay, so they get bloated and they feel uncomfortable and crampy, and that's a common problem with advanced diabetes. So, but other types of things we've already talked about. When you have your gallbladder out, or when you have bile problems, you get that fatty diarrhea, and there's a lot of things that go on with the GI tract. A lot of inflammatory, hereditary type stuff. You got arthritis in your joints. When you think about arthritis along your intestine, what that causes, that's Crohn's disease, things like that. It's an inflammatory disease where your antibodies are chewing up your intestines. Okay. But anyway, so I just wanted to show you this picture of smooth muscle. And here's also smooth muscle in the vessels, blood vessels. Okay. Remember, this is the endothelium, the simple squamous lining on the inside. The next few layers here, there's a basement membrane on all the epithelium. But after that, you've got smooth muscle wrapping around the little arterioles like this. This is what you're after with most blood pressure drugs. You're trying to get those smooth muscles to relax. Okay, so you're giving things like calcium channel blockers and blah blah blah, so they can't constrict. Okay, but some of the most common blood pressure agents work on the calcium channel. Okay, and you can see that when you're constricted, you're going to say you got a garden hose. Picture a garden hose that you've squeezed, and it's got water in it, and you squeeze the hose, well, the pressure goes up, right? Well, if you can relax that, the pressure in the whole hose goes down, okay? And think about that happening all over your body. That's what drops the blood pressure, is when you relax this smooth muscle.
But if you give someone a calcium blocker, isn't it going to essentially relax all the muscles?
Well, logically, you might think so, but
Okay.
these things are very specialized.
Very specific?
Yeah,
Okay.
yeah. You have different kind of calcium channels and different types of muscle cells and
Okay.
things like that. So not all sodium blockers work on all cells.
Okay.
Not all calcium channel blockers work on all cells. So they have to test them. Okay.
Okay.
But because you can't always get a perfect calcium channel blocker or whatever, you can sort of predict the side effects that are going to happen when it starts to work on the other cells.
Okay.
Okay, logically, you can kind of predict the side effects. Okay, but these are smaller arteries. We don't have a lot of smooth muscle, say, in the aorta. Okay, because we don't need that to constrict. Okay, but these are the small ones out in your fat and in your skin and your muscles. They control the blood flow through those tissues. Okay, this is what and you get opposite effects. The fight or flight system. When you release adrenaline into the body, adrenaline affects the receptors on the smooth muscle. It will loosen up the ones in your lungs so that you get more air into the airways. It will loosen up the ones in the muscles so you get more blood flow through the muscles, but it tightens up the ones in fat and skin so you get less blood flow, because that's not where you want it. You want it going to your muscles when you're fighting. Okay? It's a really weird system, but we'll talk a lot about it when we get to the neuro section. Okay? So don't expect a lot of questions about that right now. Just PBS. But this is why there's no striations in the smooth muscle. You have those same kind of sarcomere-like structures, but they're hooked to little protein bodies here at odd angles. So you get this fishnet sort of arrangement, and when they constrict, all it does is get a blob. The cell just constricts and looks like a blob, okay? But you don't see striations because they're not lined up in perfect alignment, okay? But you still have to have the actinomycin there for it to constrict. And you'll notice that this is probably, this is kind of a good picture here. Think about this not being one of our smooth muscle cells, but this being a single-celled animal, okay? This is probably how our muscles actually developed in the beginning. Okay? They probably look like this because remember those desmosomes? They had the connecting the fibers holding the cell together, but they had those long tails of actin. Okay? So you can almost imagine a desmosome all of a sudden getting pulled on by a walker. Okay? And you're very close to building yourself a muscle cell. Okay? That's probably how it happened, like this. And then over time, they started to align themselves up, and you get real muscle cells. But that's as close to a single-celled animal moving as you can get. A muscle in a single-cell animal. Okay? And here's your gap junctions as well in smooth muscle. Okay? And this will be the smooth muscle, say, in the intestines or in the uterus, things like that. The ones you want to move in rhythmic fashion, they're going to have the gap junctions. We do have smooth muscle that you don't want to do that, such as the smooth muscle in your iris or in your lens in your eye. Okay? You don't want to, say, look out and all of a sudden have your vision get blurry and clear and blurry and clear. You don't want waves of action like that. You want your iris to focus and let in enough light and stay there. You don't want it to be doing this. Okay? You don't want any kind of rhythmic activity. So the smooth muscle that's in the colored part of your eye, it has constrictions. And it will come down and close the iris, but it also has radiating fibers that go out, and they'll pull it open. Okay? So, and you can guess which one is controlled by what nervous system. If you're fighting, do you want to let in a little bit more light to see? Yeah. That's why people say he was wide-eyed. The way the pupils dilate when it's under sympathetic nervous system, adrenaline. Okay, the pupils will dilate. So that's the radial fibers that are pulling it open. The constricting fibers that make your eye smaller is the rest and digest system that does that. Okay, which is why a lot of drugs that work on things like stomach drugs and allergies and things like that have that parasympathetic stimulation. They'll cause constricted pupils. Okay, and people will complain about blurry vision because of that. But again, we'll talk more about that. So this is just to show you, don't worry about all this stuff here, just to show you that there are unitary or visceral smooth muscle like the gut and the uterus, and then there's other muscles that you want to control nicely, and so they actually have to have a nervous stimulation to get them to fire. They're not going to fire each other with a gap junction. Okay? It just so happens the erector pili, that smooth muscle with the hair in your skin, okay? That's controlled mostly by your sympathetic nervous system, the fight or flight system. Okay? So when it stimulates it, it fires and the hair stands up. Okay? Same reason it stands up on the back of a cat or a dog, same reason. So, but not a terribly big issue. Okay? We have, I think I'm going to do this next time because this is relatively simple, but I wanted to get to just some other things here and have a little 15-20-minute discussion. All I want you to do is to be aware that these words exist and basically what they mean. Okay? Like I said, anytime you use the word myo, you're usually talking about muscle cells. Myopathy is muscle disease, basically. Pathy means suffering. Myology is the study of muscles. And so anytime you see myo, think muscle. Muscular dystrophy. Trophy usually means trophic is changes, things that are changing. Dys means wrong or disease. So this is wrong movement, wrong changes, wrong movement basically. So that's where this group of hereditary diseases got the name dystrophy. Okay.
May I ask a question?
Yeah.
The the protein in the
Dystrophy. Dystrophin, my brother.
dystrophin, is that how it got its name because it changes
Yeah, I
the?
assume so, yeah.
Okay.
Yeah, very much so, I think.
Okay.
Yeah. It's just too similar.
Yeah.
Because they found the gene before they found the protein.
Okay.
They found the gene that was mutated because it was different than normal people.
Okay.
So they thought that had to be the reason. And then they figured out what the protein was that it was making.
Okay.
So the protein was out there in muscle cells and never been found before.
Okay.
And they can't reverse it
No, because it's genetic.
once it's already.
Every single muscle cell has bad connections, so there's no way to fix that.
Nothing done before birth.
Yeah, yeah. Which is mostly the case with almost all genetic diseases. Every single cell in your body has the same DNA, and they all have the same mutation. Okay? So it's everywhere. This is what it actually looks like. This gray area here is the cell membrane. Okay? So this is the area that has the lipid-soluble amino acids on it. This is what's going to float in all those fatty tails. So these areas have to be more water-soluble so that they stick out and function. And obviously you have to have an exposed area here. This is the structure of acetylcholine. Okay? It's vinegar hooked up to an acid with an ester reaction. So now we've got an ester. Or vinegar hooked up with alcohol, I mean. Okay, so now we have an ester. Okay, but this is acetylcholine. It sits in two places on this protein, right here and over here on the other side. And if you look at it from the top, when they're there, you can actually see the hole where the sodium rushes through. So it really is a pore. I mean, it really is a machine. It's pretty incredible. Several different proteins make it up. Okay, so genetic mutations in any one of these proteins would make this pore not work right or as well, or maybe too well. Maybe it leaks too fast or something and it causes other problems. But these were done by X-ray crystallography studies, so we know the exact shape of these things now, which is so wonderful because when I was in school, they would just write blobs. They would just write a circle and say, this enzyme does this. And we had no idea how they were working and what, it was amazing to try to learn all that stuff. But now you can actually see these things and you can see what's moving and it's just unbelievable. So, this isn't quite so pretty when you use the cartoon kind of helixes and beta sheet structures, but this is what the sodium and potassium channels look like. Okay? And I throw it out only just for amazement. You don't have to memorize what they look like. You're not going to have to identify one from the other. But this is what they are, and they really do have holes in them. And it really does matter what amino acid is right here, okay? Because that's going to be the OHs and the oxygen groups that hydrate the ions as they go through, okay? It's not going to go through, a fatty protein. It's got to go through a water-soluble hydroxyl groups and things like that, okay? So it really matters what's there. And you might mutate something out here and not affect it too much, but you mutate any of these ones where they're critical, and that channel's not going to work, and you're not going to be born. Okay, it's inconsistent with life. Okay, so and then I just wanted to throw out. This is all PBS here. Just to show you some examples of things. Anybody ever heard of tetrodotoxin? Okay. How about bufotoxin, or poisonous frogs? There it is. There it is. Yeah, this stuff will kill dogs and things that chew on them and bite the frog or lick them. My dad's dog almost died doing it down in South Florida. But tetrodotoxin is a fairly common toxin in the animal kingdom. It's come up on all sorts of different lineages here, so probably developed independently. But this is a lizard that's up in Washington and Oregon, and several people die from that every year when they fool with these things and get the tetrodotoxin gets in their skin. It's a very strong nerve toxin, and you can just imagine that thing, nice little round molecule like that, just fitting down in that hole and blocking it. Okay, and that's what happens. Okay, fitting down in that hole in the acetylcholine receptor, and because it blocks it, what happens is nothing will fire. Okay, So you don't get seizures or, foaming at the mouth or anything like that. It just blocks that pore. All of a sudden, you're flaccidly paralyzed. You can't move. Your diaphragm stops working, and you just suffocate while you're wondering what's happening. Because you can't breathe. So it's nasty stuff. Our group was even trying to synthesize that back in graduate school, so we were working on that. But the ringneck octopus, or the blue ring octopus has it. This is that puffer fish that the Japanese like to slice up and eat. That's tetrodotoxin that kills people.
There's like one little.
Yeah, sure it is.
I have Japanese friends, so that's, I live in Japan and they eat that.
If being limp and flaccid is...
They eat it because they say like it makes them a man and it's an Aphrodisiac.
Yeah, well, the risk. Yeah. But obviously they didn't get any of it if they're still alive. It's just going through the risk. Yeah.
My only experience with that is watching Homer Simpson almost die from it.
From the sushi?
Yeah. Yeah.
They had a CSI.
Oh, okay.
Yes.
Yeah, I imagine you could kill people with this, and it might be kind of easy to get away with it because who looks for this?
Right.
I'm not sure if this is on the toxin screen.
Hey, honey, I made fish tonight. Yeah.
But
You done told me the wrong thing.
what was that?
You done taught the wrong
Well, between this and ricin, we could do a lot of damage.
Just take the bag and take it back.
I'm going to keep reading
Kill your
papers now.
lover.
I'm going to make sure you guys stay alive.
Figure it
I don't
out.
want to be reading the obituaries for the next few years. So, tetrodotoxin, interesting stuff. This is curare, interesting molecule. I have no idea where this fits on the receptor. I don't know. This also causes it's a competitive antagonist. Basically, it blocks the acetylcholine receptor, okay? So it's kind of doing the same thing as the tetrodotoxin, but I don't know if it sits on the pore or somehow hydrogen bonding just closes up the protein or something like that. You could guess. But it's also a flaccid paralysis. It blocks those sodium channels from firing. So they put it on the arrows and they shoot the monkeys out of the trees. And the monkey's up there just kind of he's going, well, it's happening, and then he just falls out of the tree, and that's how they catch him. Okay? And eat him, that's what they're doing. But this is an arrow poison in South America. As opposed to the other one, the rotenone, which is a fish toxin that they use. Okay? This works pretty quickly, just a couple minutes, and they just fall out of the tree.
I wonder how many accidents take place every year with the person handling it to kill the
You
monkey.
gotta be pretty good.
Yeah, I think so.
Or the guys that are actually doing this probably don't keep a lot of records.
Yeah.
They got other things to worry about. This is sarin. This is a nerve gas. The way these things work is they attack the cholinesterase. Okay? So what happens is this is a very potent bond here. And any hydroxyl group on that protein, or a lot of other proteins actually too, is going to come up here and be sucked into this phosphorus. The electrons will be sucked in because this fluorine is pulling so many electrons off that phosphorus that makes it a positive charge, and those electrons on the oxygens will come in and hit this, and then this will leave as an alcohol. Okay, but this will permanently bond then to the cholinesterase, and probably in a lot of different places, but enough places that it shuts it down and it no longer breaks the acetylcholine in half. So now you know what's going to happen. Now you've got way too much acetylcholine in the cleft, and that's not going to stop inhibiting the neuromuscular, the acetylcholine receptor. So what are you going to die of?
Spazzing out.
The seizures, yeah. That's an excitable toxin. Okay, that's going to excite you, and you're going to die of seizures because you can't breathe. Okay? But it very often causes. That's the muscle part if you get enough of it. Okay, and very often just a little tiny bit of this, a tenth of a drop will kill you. I mean, this is really toxic stuff because even in that drop, you've got trillions of molecules. Okay, and that's enough to shut off your channels. But less toxic forms of this very type of thing is what most insecticides are. okay? They're acetylcholinesterase inhibitors. That's what kills the wasp and kills the flies and kills, you know, it just shuts down their nervous system. And you've probably seen them shake. Well, that's what's going on. And when kids, they're less toxic than this thing, but when kids get out in the garage and get a hold of that stuff, they'll get poisoned with this. Not always to the point that they're going to die, but they get what's called a sludge syndrome. I think I might even mention it. That's because this is turning on all the acetylcholine nerves, and that's your parasympathetic nervous system. That's the rest and digest system. So you're going to get the salivation for digestion, okay? You're going to get lacrimation because you don't want to be crying when you're fighting, so it's the opposite, okay? The parasympathetic gets the tear glands. So you're going to be frothing at the mouth and watering in the eyes. You're going to be throwing up. You're going to be overstimulating the intestines. You're going to get diarrhea, cramping, abdominal pain. That's the sludge syndrome. Okay?
Is
Yeah.
that found in like lye?
No, lye is just sodium hydroxide.
Because my dog, we put lye out for the police, and my dog, everything that you just explained with that sludge syndrome, she had that.
Well,
I gave her milk and she came back around
Yeah.
after about a couple hours, but
well, lye is, like I said, is just caustic soda. That's sodium hydroxide or potassium hydroxide. But take a mouthful of that, see if your mouth doesn't water.
but I mean she was defecating and she was like, and I picked her up and then I tried to like stand her up and she just like she couldn't do anything.
Wow. Yeah.
Yeah, it was really weird.
But it's definitely not a nerve toxin. That's it'd be like drinking acid. You know, you're gonna get a lot of symptoms. Okay. Atropine. Everybody remembers the angel trumpets? Yeah. When I first moved here, I used to hear about, you know, all the teenagers making tea out of it. It was a big deal. Some of them had died, and you know, and others were just freaking out in the ER. They thought they were, you know, manic depressive episode or something. But it was a big deal. I haven't heard about it much recently, but But a student in my other class said she's got a couple of in her yard, and she said once they flower, the next day they're gone. So people are coming by and pulling them off.
Wow.
But this makes atropine. Okay, you've all probably heard of atropine. It's used medically a lot. What it is, it's a blocker of those same cholinesterase channels. Okay, if you get too much of it, you're going to get all the symptoms of a flaccid paralysis and, you know, brain problems. But because there are some acetylcholine receptors in the brain as well, believe it or not. But this is used as an antidote in small dosage. Okay, it's for this poisoning. Okay? Because if this problem gives you too much acetylcholine in the cleft, what do you want to do? You want to block the pores so the acetylcholine doesn't have the effect that it would. Okay, that's why soldiers carry around atropine injections when they're considered being in danger of nerve gas attacks. Okay, and it's also the kind of thing you give kids that have poisoned themselves with the acetylcholinesterase inhibitors, insecticides. So, and because it blocks the acetylcholine receptors, it also dries you up. So it's used during surgery to keep your saliva down and keep your eyes from watering. And, you know, it calms down the GI tract because it blocks all the effects of the rest and digest system. Okay. So that's why they use it. It blocks the effect of the vagus nerve, which would slow the heart down. So that's why you give atropine during cardiac arrest to speed it up. Okay. So then you give adrenaline, so that it's like you're taking over the job of your fight and flight and rest and digest system. The vagus nerve will slow the heart rate down. Adrenaline will speed it up. Okay. So what you want to do is block the vagus nerve acetylcholine receptors and give adrenaline to speed up and stimulate the heart. That's part of the code drugs. Oleander. Poisonous. Very poisonous. Don't let your dogs chew on them if you got them.
Is that the way it's poisonous, by
The sap.
ingestion?
Yeah, the sap. If you chew on the leaves or the stems. A lot of people don't know that that's as toxic as it is. As a matter of fact, it was like the first week when I moved here 25 years ago, I read a story about a family in Daytona. They were here on break, and Holiday, I guess they didn't know about it. They were doing a weenie roast using sticks of oleander, and so they're, you know, heating up that wood.
See, I had heard about that. We had oleanders, and I wasn't sure if it was the burning or the actual, you know, the limb itself somehow ingesting it.
Yeah, I think you got to ingest it, but if you burn it and breathe it in, I can't imagine. I haven't heard of anybody getting.
Yeah, that's what I wasn't sure. When I heard about that quite a few years ago, probably about 10 to 15 years ago.
It might still be toxic. Who knows? You know, I just haven't heard of it. It's just like burning poison ivy. You know, that smoke
bad
is still
too?
toxic. Yeah, you'll still get poison ivy if you get that smoke on you.
Oh, I didn't know that.
What kind of molecule is that?
A steroid?
You got it.
Yeah.
6,6,6,5. And that's just a sugar. Okay. So the plants is sticking a sugar on a steroid, and these probably comes from another sugar type moiety, but interesting structure. And so you can't always say that, you know, steroids are good for you. But look at the symptoms now. I have no idea. I'm not even sure it's known. I think I just took this off of Wikipedia or something. They didn't say anything about what receptors were being worked on, so they may not know yet. I don't know. But look at the symptoms.
Yeah.
Nausea, vomiting, salivation, abdominal pain, diarrhea. What do you think it's doing? Esterase? It could be.
Esterase,
It
yeah.
could be acetylcholinesterase, just like the nerve toxins. Okay. Either that, or it's stimulating those receptors and causing them to fire, just like acetylcholine does. Okay. So one or the other, it has to be, because it's a stimulator, not a flaccid paralysis type of thing.
Right.
4 minutes. This will be enough. We'll stop here. Chiguatera. Anybody ever got this? Anybody doing deep sea fishing in the islands or anything?
That's a crazy molecule.
That is a hell of a molecule. I'm sure somebody or 5 people got their PhDs just figuring out the structure of that thing.
Wow.
Because that would be tough even with modern techniques. But how that wraps around the receptors, who the heck knows? You know, I don't know. But look what it does. It lowers the threshold for opening the voltage-gated sodium channels. Okay, you would think that would give you numbness, right? If you go from -70 to say -80 or -90, it takes more to get up to threshold, so it's not going to fire as well. So I would expect things like paralysis and numbness, things like that. Okay, the nerve, the sensory nerves may not be working. But it does some weird things. It does some of the vomiting and diarrhea as well. But their mouth and lips, numbness, that's exactly what I would have expected. Okay. But this, I have no idea how this would happen. Reversal of hot and cold sensation. Okay. So what I assume they mean by that, and I haven't asked anybody, but you put your hand in ice water and it feels hot. Reasonable.
That's what
Yeah.
it sounds
You
like.
put it in hot water and it feels cold. Okay. I don't know how that would happen.
But you were telling us that there's, or if I remember correctly, that there's different genes that control.
2 different types of hot and cold nerves. Right.
So could it just be attacking?
But how could it just reverse?
I don't know.
I don't get it. Okay, so I'll take their word for it, but I don't understand it. I'll have to get my PhD working on that.
I don't think I want to try it and find out.
No.
Last night, there were a couple people from the islands, Jamaica, one of my students said his dad's had it about 5 times.
Wow.
He gets it from Barracuda.
Oh, man.
He said he eats maybe 1 Barracuda every month, you know, so he's had hundreds of them over his lifetime, and at least 5 times he's got Chiguatera. And he said it was really awful sensation. It's kind of like pain in your joints and numbness and tingling in your hands. He said that his hands in his mouth, and he said, and he was on speakerphone, and he's going, and I just couldn't have sex because my wiener was burning. Oh, it was
That's awesome.
What do they do so he keeps eating it? Like, how is it?
As far as I know, there is no treatment for it unless somebody.
Just like go out of your system.
Yeah, it takes weeks. It takes time. Yeah. I'm pretty sure my brother got it a few years back when he was doing some gold diving out in the Dominican Republic. And the whole ship, there was about 5 guys that got sick and had the similar types of thing, a lot of numbness and tingling. Not a bad case, and they went away in a few weeks, but I think that's what happened. Well, I wasn't aware that it's as common as it is in the islands, talking to that student. He said, yeah, all my relatives have had it. And I said,
Yeah, because grouper is a wide fish,
Yeah.
they eat there too.
I think barracuda is the one they always blame though.
Yeah, I've had very.
It's the predator fish. It's like mercury gets, goes up the line, the more little fish get eaten. The big predators get all the mercury in the end.
Right.
Well, it's the same way with this toxin from this dinoflagellate. Okay? It gets in their fat and it just stays there. Okay? So it's just a matter of luck. I mean, you can't avoid it.
No.
If you're gonna eat those fish, you're taking a chance.
So is that, from the mercury you saying?
No. I'm saying just like mercury,
Oh,
it
okay.
accumulates in predators.
Okay.
It goes up the food chain. Okay. Thank you.